Hi guys, welcome back to another video in Microbiological Insights, and today in this video we will be going to see about the very important topic in microbial metabolism, that is about the glycolysis pathway. And this glycolysis pathway is the best example for anaerobic metabolism. Let's move into the topic. What is glycolysis? Glycolysis is the metabolic pathway which is used by most autotrophic, heterotrophic microorganisms, aerobes and anaerobes to begin to break down glucose. So here the organisms break down the glucose molecule. What is an autotroph? Auto means self, no? So those organisms which can produce their own food. What is a heterotroph? Heterotrophs means these are the organisms which depend on other organisms for its food. Aerobe means those organisms which lives in the presence of oxygen, and anaerobe means those organisms which lives in the absence of oxygen. So these all group of organisms begin to break down the glucose molecule. The name glycolysis means, glyco means sugar, lysis means splitting. So splitting of sugar molecule. And this pathway does not require oxygen molecule, but it can occur in the presence or absence of oxygen. And this glycolysis pathway is otherwise termed as Emden, Meyerhoff and Parnas or EMP pathway. And these are the photographs of Gustav Emden, Jacob Parnas and Otto Meyerhoff. They only discovered the glycolytic pathway. So only the glycolytic pathway is otherwise termed as the EMP pathway. And this is the structure of glucose and fructose molecule. If we know this structure very well, it is easy for us to understand the pathway. When we see the glucose molecule, it is a 6-membered ring structure. The number starts from 1, 2, 3, 4, 5, 6. And when we see the fructose molecule, it is a 5-membered ring structure. The number starts from 1, 2, 3, 4 and 5. This glucose and fructose is a monomer, no? These are also isomers. What is isomer? It have the same molecular formula but different functional group. The molecular formula of both glucose and fructose is C6H12O6. But glucose have aldehyde as their functional group and fructose having ketone as their functional group. That is the only difference. And let us move on to the steps of the glycolytic pathway. The glycolytic pathway have 10 steps. And in these 10 steps, mainly 4 important events occur. What are the 4 important events? The first one is the substrate level phosphorylation. That means the transfer of phosphate group from ATP molecule to glucose. So in the substrate level phosphorylation, the phosphate group is transferred from ATP to glucose. And the second one is the breaking of a 6-carbon molecule, because glucose is a 6-carbon molecule, no? So this 6-carbon molecule is broken down and changed to 2 3-carbon molecules. And the third one is the transfer of 2 electrons to the coenzyme NAD. And the fourth one is the capture of energy in ATP molecules. So the energy is stored in the form of ATP here. The first 5 steps comes under the preparatory phase, and the last 5 steps comes under payoff phase. Let us see each steps in detail. Step 1. Phosphorylation of glucose. In the first step of glycolysis, phosphorylation occurs. And this is the first priming reaction. In this step, the first ATP molecule is utilized. So what happens here is, this is the structure of glucose molecule. The glucose is first phosphorylated at the hydroxyl group on the C6 carbon. So the C6 carbon molecule have a hydroxyl group. And so what happens is, this H is removed, and so one phosphate molecule is added here. And the structure is now like this. The only change is the H is removed and P is added here. The process involves the transfer of phosphate from the ATP to glucose. This phosphate comes from the ATP molecule. So the ATP donates its phosphate group to the glucose molecule and it is added in the sixth position. This reaction is carried out by the enzyme hexokinase. And during this reaction, a considerable amount of energy is lost in the form of heat. In the first step, the glucose is converted to glucose-6-phosphate. First ATP molecule is utilized here. Let's move on to the step 2. The step 2 is the isomerization of glucose-6-phosphate. The glucose isomer is converted into fructose isomer here. Glucose-6-phosphate is reversibly isomerized to fructose-6-phosphate by the enzyme phosphohexoisomerase. What happens here is reversibly isomerized to fructose-6-phosphate. You can write the structure of fructose here and adds the phosphate group in the sixth position. So it's very simple. This reaction involves a shift of the carbonyl oxygen from C1 to C2. What happens in this reaction? In this reaction, a shift occurs in the carbonyl oxygen from C1 to C2. The oxygen is shifted from the first position to the second position. The structure will be like this and combine this together CH2. One oxygen OH hydroxyl group and another hydroxyl group. So you can add these two hydrogen molecule together CH2. OH here, OH here. The aldose molecule is converted to a ketose molecule. This reaction is carried out by the enzyme phosphohexose isomerase. And now the glucose-6-phosphate is converted to fructose-6-phosphate. Let's move on to the step 3. The step 3 is the phosphorylation of fructose-6-phosphate. And this is the second priming reaction. So what happens in this reaction? The second ATP molecule is utilized in this step. You can write the structure of fructose-6-phosphate here. And the fructose-6-phosphate is converted into fructose 1,6-biphosphate. Two position having the phosphate group. First position and the sixth position. So this is the first position. This is the sixth position which is already having a phosphate group. Remove the hydrogen from the hydroxyl group and add phosphate. So the structure will be like this. This reaction is carried out by the enzyme phosphofructokinase. The ATP donates its phosphate molecule to the fructose 6-phosphate. Here also some energy is lost in the form of heat. The fourth step is the phosphorylation of fructose 1,6-biphosphate. So here what happens? A cleavage occurs in the CC bond of fructose 1,6-phosphate, third and fourth position. So it leads to the formation of two different triose molecules. The first one is the glyceraldehyde 3-phosphate and the second one is the dihydroxyacetone phosphate. Now you can write the structure here. POCH2, then you can see these three things, OH group, H. So total 3 H is here and 2 O group is here. So you can add one keto group, C double bond O. Again the remaining is 3 H. So CH2, 1 O is remaining, so OH. So the structure will be like this. This is dihydroxyacetone phosphate. And now we are going to write the glyceraldehyde 3-phosphate. The structure will be POCH2. POCH2, then CH, OH. Remaining is H and O. So one ketone group, C double bond O, H. This is aldehyde group. So glyceraldehyde 3-phosphate. The remaining steps in glycolysis involve 3 carbon units rather than 6 carbon units, because till now we see the structure as 6 carbon molecule. No, here now the remaining steps having only 3 carbon units. And the fifth step is the isomerization of dihydroxyacetone phosphate. So isomerization means it can change from one isomer to another, no? The glyceraldehyde 3-phosphate can be readily degraded in the subsequent steps of glycolysis. But dihydroxyacetone phosphate cannot be. So the dihydroxyacetone phosphate cannot be degraded easily. So it can isomerize its structure into glyceraldehyde 3-phosphate. These are the structures of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then the dihydroxyacetone phosphate again changed to glyceraldehyde 3-phosphate. So at the end of this step, we have two glyceraldehyde 3-phosphate molecules. And this reaction is carried out by the enzyme triose phosphate isomerase. Up to this step, the first phase of glycolysis completes. We move on to the second phase of glycolysis. Step 6. Oxidative phosphorylation of glyceraldehyde 3-phosphate. This step is the energy conserving step or energy forming step. So here some energy is going to form. Here the glyceraldehyde 3-phosphate is converted to 1,3-biphosphoglycerate. So you can write the structure as POCH2CHOH like that same. The only change is remove the H and you can add O and P. 1,3-biphosphoglycerate. And this reaction is carried out by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, NAD is reduced to NADH. These are the coenzyme, no? What is NAD? Nicotinamide adenine dinucleotide is converted to nicotinamide adenine dinucleotide hydrogen. Since two molecules of glyceraldehyde 3-phosphate are formed from one molecule of glucose, two NADH are generated in the step. So here two glyceraldehyde phosphates. We get two molecules of NADH. And this is the 7th step, the transfer of phosphate from 1,3-diphosphoglycerate to ADP. It is also the ATP generating step. Here the transfer of phosphate group from 1,3-biphosphoglycerate to ADP. So the phosphate moves from the 1,3-biphosphoglycerate to ADP molecule and it is converted to ATP. The reaction is carried out by the enzyme phosphoglycerate kinase. So since two molecules of 1,3-biphosphoglycerate are formed from the one molecule of glucose, two ATP are generated in the step. So here two molecules involved. So we get two molecules of ATP here. And the 8th step is the isomerization of 3-phosphoglycerate. Isomerization occurs in this step. So the 3-phosphoglycerate is converted to 2-phosphoglycerate with the help of the enzyme phosphoglycerate mutase. So now see the structure. POCH2CHOH C double bond O. This is the structure of 3-phosphoglycerate. It is converted to 2-phosphoglycerate. You can shift the phosphate group here. So it is CH2OH. You can add hydrogen here. So CH2OH and this hydrogen is shifted here and phosphate is going to shift here. Change the structure like this. CH2OH, here CHOP, the remaining are the same. This step is a reversible isomerization reaction. 9th step is the dehydration of 2-phosphoglycerate. So what happens here is 2-phosphoglycerate is converted to phosphoenolpyruvate with the help of the enzyme enolase. You can see the structure here. 2-phosphoglycerate is converted to phosphoenolpyruvate. So OH is removed here and the bond, double bond. You can see the double bond here also double bond. The remaining is the same. Here two molecules of water are lost. And the last step is the transfer of phosphate from phosphoenolpyruvate. And this is the second energy generating step. So second ATP molecule is formed here. In this step, the 2-phosphoenolpyruvate is converted to pyruvate. What happens in this step is CH2 is here you can see the double bond. So it is converted to single bond. So CH3. Remove the phosphate group and you can add the phosphate group to ADP. So it is C double bond O, the remaining is the same. So here the ADP is converted to ATP. The reaction is carried out by pyruvate kinase enzyme. The keto form predominates at pH 7. One glucose molecule is converted to two pyruvates. When we see the yield of ATP molecule means two ATP molecules are used in the 6-carbon phase to form the fructose 1,6 biphosphate, and in the second phase total 4 ATP molecules are generated. So we can subtract the ATP molecules used in the 6-carbon phase means we have only 2 ATP molecule. Glucose, one glucose molecule plus 2 ADP plus 2 phosphate plus 2 NAD gives 2 pyruvate molecule, 2 ATP generated here, 2 NADH generated plus 2H. So this is the overall reaction. And now we are going to see about the overview of glucose metabolism. In the glycolysis pathway, the glucose is converted to 2 pyruvate molecule. So again this 2 pyruvate molecule is converted to different forms in anaerobic fermentation and aerobic oxidation. Under aerobic conditions, the pyruvate formed by glycolysis is further oxidized by the citric acid cycle. So this is the aerobic oxidation. So this pyruvate is further oxidized via the citric acid cycle and oxidative phosphorylation to yield carbon dioxide and water. So 2 pyruvate molecule is converted to carbon dioxide and water. And under anaerobic condition, two types of fermentation takes place: homolactic fermentation and alcoholic fermentation. Here NADH is reduced to NAD and the end product is lactate. And in the anaerobic alcoholic fermentation, the final yield is ethanol and carbon dioxide. So this is the overview of glucose metabolism, and these are the reference book. I think you understand very well about the glycolysis pathway. So if you find any difficulty in these steps, you can write your doubts in the comment section. Okay, thank you.
If you like this video, please don't forget to subscribe, share, like and click the bell button. If you have any more doubts, you can write your doubts in the comment section below. Thank you.